Thank you, Catherine, if you can hear me. So, Gerald, first of all, you are absolutely smart today. I mean, you always dress very well, but I've never dressed. Given what I think of your dress sense, I've never thought I'd ever see you in a suit. So, I actually just have a couple of questions, Gerald. So, these specific loci that you identified, which is absolutely amazing, because again, it's a new population, and like Kathy referenced, this is an extreme phenotype. So that means that a lot of potential samples were left out for the right reasons, because the design is extreme phenotype. But I wonder then, how then you are able to measure the predictive power of these loci. So I know you did GWAS, and in GWAS you can see what sort of blocks to the top. And I guess this is how you identified them initially. And you have p-values for that. Yes. But I wonder, especially given what you referred to, that it is a complex, it's a complex phenomenon. What your thoughts are on how one can effectively measure the predictive power of this? Because so you have this sample and you have this loci that you've identified, but what you see is really a statistical signal. So there is, it's possible that there are some of these children that have these loci but have the other unexpected phenotype when you dig down to the individual children. And so what you see when the thing stands out from GWAS is a population level statistical thing that pulls up. So I wonder what your thoughts are on how you could measure the predictive power of these loci.
So thanks, Dr. Daudi, for that comment. What I can say is, it's unfortunate that the study design and the cutoffs for these calculations were made based on a very highly homogeneous population like the Caucasians, and they cast these cutoffs on a stone. If you are to publish a paper, they want anything less than 2.5 times 10 power minus 6. And yet we know Africans are extremely, very genetically diverse. So attaining that power is extremely rare, very rare, even when you're using the best study designs. So what I can think is, maybe it's time for the African scientists also to think of moving the bar and decide what will be the cutoff among the African population. Because like you say, you put this bar so high, you find signals are not attaining this statistical significance, and then you say the story is ending here. Yeah, so that is not good, but what I can say is you can still move beyond that point and then try to look for a different way of interrogating these variants that are not attaining this power, but interrogate them in some other way, using some other tools. That can be done. Personally, I think I can do that even in this data set. It may be my next step.
This is where my thoughts are going, because you know, if even with the data set as it is, and with the bar as it is where it is set, accurately or not, it is still possible to look in the current scenario, to look at these variants and say that with this data set, if I get a child with this genotype, then the predictive power is 80% or 70% or 50%. And I think that would would properly parameterize this. So I think that would be good work that you could follow it up with. And then I have just one other question. I think Kathy did a great job of pulling out a lot of the things that stood out. But again, this is with extreme phenotype. So you define children as which age range?
For the rapid progressors, we are picking those below 3 years. And then, progressing to 8 within 3 years, but again, we pick those as well on the extreme.
Okay. Yeah. And then for the long term?
7 years and above, then we pick those as well. Like 12 years, 13, 14, that was the extreme. I think the oldest was about 16 years.
Okay. And long time non-progression was defined as
7 years was the cutoff, moving.
Okay, and so somebody is classified as a long-term non-progressor if they spend how many years?
7 years.
Minimum. 7 years.
Yeah.
Okay. Because, and a related question to Kathy's thing then is about how many children in your cohort were, above 12 years or above 13 years?
So in the combined cohort, you had 141 were LTNPs.
Okay.
And then we had 173 as rapid progressors.
But that age distribution in the LTNPs, about how many were 12, above 12 or above 13 years?
I don't have it of it, but I think it was a small number, much as it is extreme. But I remember the oldest was 16 years, but there are not so many who are above. There are not so many who are above 12. There are not so many.
Okay. I think that answers it, because if there are not so many, then it wouldn't skew the results very much. If they're significant, it would be a little bit of a problem, because by the time they make the cut, they're out. If you're taking 3 or 4 years as the period for somebody to be qualified as a long-term non-progressor, if they're 14, then by the time they come out, things have changed. So, but if there are few, then that's it.
Yeah, there are few.
Well, thank you very much, Gerald.
Thank you so much, Gerald, for such a wonderful presentation. I think Kathy and Daudi have been up there. I want to come down so that I can understand, because I'm from mathematics. Now, you have talked about one of the proper usage of this research as being a potential therapeutic targets. And you said that for HIV we have about 5 different drug types, and you mentioned some of them. But for the other 5, they have majorly targeted the life cycle of the virus. How do you go about this, in case you found something interesting?
Okay, so what I can say is, my work was looking at developing a screening target at the host level. It is easier to have a target, a therapeutic target, against a small virus such as HIV, which is 10 kb. A human genome is 3.2 billion base pairs. So the rationale why they are interested in targeting the virus, it is too small. You have a small target you're dealing with. But now if you found a marker on this other side, for people who have seen the CCR5, I think it's a miracle mutation that of late, I think there are 2 people who have been clinically cured of HIV using the same approach of CCR5. So, fine when you are using if you are looking at the target in a virus, still we can get a drug, but the drug may not be in position to wipe away the disease. But the target on the side of the host, the benefits of having a target on the side of the host are so high that still, like I've said, the CCR5 is a miracle cure. With so many advances, we can't cure the virus and so on, but when you deal with the host at that level, you can sterilize the host of the virus. That's what I can say.
And my second last question is, when in around 2008, 2007, people were thinking about such studies, or were doing these studies, the thinking then was that Africa was one continent with a very uniform genetic makeup. Now we think it is not true. But now what we are saying and what you have said is that if only we had a tool that was specific for the African population. And you have given us a great hope that the H3 Africa chip might go a long way in addressing some of the shortcomings. But I'm even worried, is Africa really, will that one address the issues that we're seeing in terms of the data that we're getting out of Africa? Looking at Uganda as an example, with migrations from the south, from the north, from the east, all different makeup, with the one Ugandan chip be able to do it, leave alone Africa.
That's a hard question, but I'll try. Yeah, so the diversity on the African continent is big, like you have said. I think the last time I checked in Uganda, we have more than 70 tribes. Yeah, so you can't ask me and I name even 20, to be sincere. But when you check the books, we have 70 tribes. And then it's unfortunate, but you can't wake up and say people are only married within their tribes. So what I can say, the human genetics is trying to interrogate it is more of like shooting a moving target. So the target is moving and you're shooting at it. So I don't know how far you can go when you're dealing with a scenario which is presenting itself as such.
So Gerald, so I mean, related, where John is going is that this is partly a logistical limitation. So the amount of genetic diversity, even within Africa, with its diversity, is still finite. It is still finite, and so if theoretically you sequenced every African, the chip that you manufacture will capture all the diversity. So this is where John is going with that question.
And just looking at the slide that is up there, you can clearly see that first line, the Ugandan, looking at the Ugandan variants and the combined, even just looking at the Botswana population and Ugandan population, you need two different kind of cases.
Part of the reason Catherine was really interested in this slide, Gerald, is it's a really an amazing number. When you see 831 for Uganda and 524 for Botswana, that's more than 50% difference. And the difference you see there is bigger. It's way bigger than if you got somebody from Sweden and compare them to somebody from Italy. You would have a difference of about maybe 20,000. Here you have a difference of about 300,000. So it's a lot. It really means we have a lot of work to do. And thank you for having this slide.
It was following up on that point. You could have even been more emphatic on the conclusions that then you draw from this result, because it is amazing. It can clearly show what the shortcomings of doing genomics in Africa are and what we need to do in order to try and start to get very interesting. And finally, you said something which amazed me when you were talking about TB and HIV. I don't know whether I got you right, that you said if you looked at the genome of TB and HIV, you would expect to find a lot more variation about the TB, because the TB is a bigger genome than the HIV. Is that correct?
Yeah, the TB is 4.4 million base pairs.
No, in terms of variation.
Okay, so if you had to keep and say, okay, every 200,000 base pairs of these genomes, how many variations am I finding? So if you had to do it at that scale, HIV is more valuable than, or have more variation in HIV than TB. But I was discussing it at the totality, that you have a bigger genome, and if you have to try to look for variation, you're likely to do it.
And just to find out for this particular study, you have access to the viral data?
I've not, but I know if I request for it, it can be.
From what Cassie has asked and from what I can see, is that I think there is a lot to be learned. If you could analyze the human and the viral data together, for example, the question that she raised of the question of why we see a lot more uniformity and why we are able to discover things in the Caucasians and we're not able to do it amongst a study like this one. So looking at just the viruses from these two populations, like you said, would lead to a very interesting study if you are looking out for that. But this is very good work. This is pioneering work, and I think the sky is now the limit for us, and the project, the supervisors and the students have laid a good foundation for us to work with.
Thank you very much.
Yes. Thank you very much for that presentation, Gerald. And I'll share all these sentiments. It's like I'm talking to somebody different, which is to. But first off, let's go to your problem statement. I want to start from there. So, no, it's the next slide where you say that this. The next slide.
Next paper. What have we said?
Maybe I've missed it, but I thought it was in the problem statement where you mentioned that this would be representative of sub-Saharan Africa.
Oh. Okay.
And that's where I had the issue of, throughout your thesis you were talking of Uganda and Botswana.
Yeah.
However, when you come to Uganda, like you said, we have over 70 different tribes. Yeah?
Yeah.
And so you didn't really break down the ethnicities, which would explain why you're seeing some diversity in there. The ethnicities within each of the countries. Because if you came to this room, we are all Ugandans, but if you did a PCA plot of whoever is in here, and you've sequenced them, you will be surprised. You will really be surprised. So, I wanted you to actually break down the Ugandans. Were they a homogeneous population of the same ethnic group, or were they individuals of different ethnicities? Because I kind of do, my analysis that I do has shown that there's a high correlation between the ethnicity and the genotype that you have.
Okay.
So I was asking whether you actually did break down to the actual tribes that are in those.
So that's very important. I remember maybe 3 years ago when you are presenting this work to the IRB, the question was, so when you are doing the analysis, will you turn to the tribe and say, okay, let me analyze the virus and then the progression at the tribal level? And I said yes, then Dr. Mark said, you, watch out. So when you look at the paper in the African Journal of Human Genetics, it was okay to analyze the clustering at the tribal level. And what I can say in this sample size we used, we represented 20 tribes from Uganda. 20 tribes, yeah. I have the breakdown of numbers, how many we have at Teso, how many we have at Uganda, but
yes.
I cannot zoom beyond there.
Exactly, and
Yeah.
I noticed that you didn't give good justice because the paper that you showed in your objective 2, Haone's paper, the Botswana individuals, he kind of broke down the population structure there before he went into the further analysis that he did. And for this case, in that objective, I thought you would actually give us some substructuring among the individuals that you sampled before we go into the actual functional analysis of identifying those particular hotspots.
And I just referenced it because the honest paper, they did the.
But they didn't do the Ugandan cohorts. They
But
didn't
it's
really break it down.
supplementary.
It's supplementary.
Yeah.
Okay, that's clear. Maybe if I can rephrase this, if you are to redo this study, would you still target the exome?
No.
No. Okay. And if you are to compare it with, Kathy highlighted it, but I wanted it to come out more broadly, that if you are to compare it with the whole genome sequencing, what more information would you get that you're not getting from this exome? Because I think when the study started, you were like, it's exciting to just target the functional loci, happening in exomes, I'm targeting these variants, they affect the protein function, and I can really see a phenotype. But I was really surprised when you didn't get anything, I was really, really surprised because it was targeting something functional as compared to the whole genome, where you have a lot of things that might not affect. So how? So
looking back in this slide, you remember somewhere where Kathy said you have 12% represent the exotic variation.
Yes.
So I mean, right from the start, you are best. You're using a capture kit that has been developed on a Caucasian population, and it comes in to pick 12%. And it is only 1% coding percentage in the human genome. So there are so many points at which you are losing the signal you would have detected. That's why I'm saying, give me money, I can go whole genome and then I'll pick whatever.
So my final question is about the chemokines. Kathy mentioned it because you had already done the whole exome, genome-wide, and you saw there was nothing, and then you went down to the chemokines. But then I read that when you're doing the validation, you did some kind of SNP genotyping, right?
Yeah.
When you did the validation. So I was really curious why you didn't do a candidate gene approach for the chemokines. You know, you had the population, you just sequence those candidate genes and then identify the loci. I thought you would have done it, but why didn't you? You're able to validate the SNP genotypes?
Yeah, we did that.
You did SNP genotyping
Yeah.
for that particular
Yeah,
SNP?
using Tuckman.
Using Tuckman, but then I thought you would actually go for the candidate genes, those chemokines.
Yeah.
Since you hadn't seen them in the genome-wide, I thought, and publications have shown that, you know, they are significant for tearing apart, teasing apart these phenotypes. I thought you would do a candidate gene approach where you amplify those genes, genotype, just that gene fragment, and then analyze that. I was just curious why you did have that approach.
Personally, I've not come across a panel for chemokines. I've not come across that.
Not the panel, you amplify the gene and just sequence.
But there are many.
Yes, you can even do some sequencing.
Because these are like-
population.
You have delta, beta, gamma, and alpha chemokines. And to be sincere, they listed more than 100. So you look at the chemokines and their ligands. So the list is endless, unless there's a capture panel that is enriched to pull from the DNA samples. It only pulls the enriched, or it only pulls out the chemokines. But I have not come across a panel that only pulls out from the DNA, it only pulls out the chemokines. I have not come across that. But if you are doing individual PCRs, I think it is too much work. Yeah. It's too much.
Okay, my final question is about the LD linkage disequilibrium analysis.
Yeah.
It was one of your objectives, but it
Yeah.
did kind of come out so well.
Okay, so that I did in a dissertation, but here what happens, I was interested in having a target, a signal that reaches less than 0.25 times 10 power minus 6, then do LD around that. But since I didn't have any, so it wouldn't make sense. But in a dissertation, I tried to do around the statistically suggestive and still see what was flanking it on either sides in a region of about 500,000 base pairs.